Professor. Thank you for your presentation. Now we have questions from our participants. The first one is hair loss when we got stress related to autophagy, like the body doesn't want to spend more energy since energy is more required in other tissues?
I don't know anything about that. I know that autophagy is involved in a lot of stresses, but I don't really know anything about that topic.
Okay, thank you. The other one is, thank you for the explanation, Professor. Is the inhibition to the release of the virus by the SNAP47 significantly decreasing the phytogenicity and infectivity of the virus in the infected organism?
Yeah, we certainly think so, and we don't know much about SNAP47 in vivo yet. We don't have a SNAP47 knockout mouse. It's been very difficult to make knockout mice to some of these pathways because they seem to be so essential. If you don't have the ability to fuse membranes together, it's really difficult, especially just the development of the mouse. There's so many membrane changes and membrane events that occur that we actually don't, we don't know what's going on. So SNAP47, we're really excited about it because it's so essential to autophagy, and it seems so essential. We lose some of these proteins and we lose 1 log of virus, and we lose SNAP47 and we lose 2 logs of virus. So it's incredibly essential. We do have now CRISPR knockout cells. All of the data that we did with transient knockdowns is holding up in the knockout cells. And so, we're right now working on understanding the pathogenesis and the amount of release we see in these cells.
Thank you. Next one is, what do you think about the development of the RNA vaccines this year? What more can this development show us?
Yeah, so it's a very exciting technology. It's something that I would say, if you had asked a group of virologists 1 year ago, what's the vaccine going to look like? Which vaccine is going to work? And I think all of us, maybe there are some exceptions, but I would say many of us, and certainly the people I talked to, would not have bet on the mRNA vaccine, simply because it was an unproven technology. Simply because, although it theoretically was sound, the idea of going from something that had never been a successful vaccine before to something we would deploy against a new global pandemic was just too much to imagine. So most of us thought that the vaccine was going to wind up being a more traditional vaccine. I think the second bet was something like the adenovirus, based vectors, like we're seeing, from some of these groups. But the mRNA vaccines that have come out from Moderna and Pfizer would not have been our first bets, just because it was such a new technology. And the fact that it's worked so efficiently, and the fact that the global pandemic spurred us to develop this technology at a large scale very quickly, means that now it's going to be accessible for all sorts of things, and it's going to accelerate the development of vaccines against all sorts of pathogens enormously.
Thank you. The last question is the SNAP proteins protecting the neuron cells from virus attacks, or the low variety of viruses that attack neurons?
Yeah, that's what we're really interested in looking at right now. And what our biggest issue, to be honest with you, is a technical issue, which is that our viruses exclusively infect human cells. And this is important to think about when you're thinking about your own projects and developing your own work. It turns out that most neuroscientists don't work on human cells. They work on rat neurons or mouse neurons, and those aren't really useful to us because our viruses are, poliovirus won't infect a mouse cell unless you have transgenically given it the poliovirus receptor. So we would have to essentially develop our entire own set of processes. So we've been trying to look at good models for human neuron culture, and it's very, very difficult. So we've now developed processes by which we're using iPSCs to develop our own neuron-like cultures. We're also using cells called SHSY5Y cells to look at neuron cultures. So now that we have those models established, to get to your point, we're trying to knock down SNAP25. We're trying to look to see if SNAP25, which is the neuron-specific SNAP, is protective in those cells. And it's a great question. And it's just the reason I can't answer that, and Chung has been working on it in my lab, and now I have another person working on it, is that it was just a huge technical challenge to find a good human neuron model, because the people who are just interested in neurons, it's much easier to get primary neurons from a rat or a mouse, which aren't helpful for us. So we're looking at it. We don't have an answer yet, but it's exactly the question we're asking.
Dear Professor, thank you for your informative presentation and answers. And also, thank you for participating in our event. It's an honor for us to listen to you.
Well, thank you so much. And it was an honor to be asked. I really appreciate it.
Dear participants, now we have a break.